Dr. Gruber, are you ready to roll?
Sure.
Wonderful. Welcome.
Thank you.
We are absolutely delighted to have you. Dr. Staci Gruber is the director of the Cognitive and Clinical Neuroimaging Core at McLean Hospital's Brain Imaging Center and an associate professor of psychiatry at Harvard Medical School. Her clinical research focuses on the application of neurocognitive models and brain imaging to better characterize risk factors for substance abuse and psychiatric conditions. Her laboratory has generated numerous publications on both recreational and medical cannabis use, publications that have been enormously influential to the field and have been cited hundreds and perhaps even thousands of times at this point. For more than 2 decades, she's been a leader in the field of cannabis research. So please join me in welcoming Dr. Staci Gruber as our first keynote presenter for the conference. Thank you so much for joining us.
Well, thank you. And first of all, let me just say this. I am a huge fan of your center and all of your efforts, and congratulations for what sounds like an incredibly productive time amidst what we call the craziness here in the US, which of course could refer to any number of things here in the US. I'll leave it there. Let me see if I can share my screen, because I bet you're going to want me to do that. Let's see what we can do here. But really, it is an honor to be here, and I'm delighted to kick this off. You guys have an extraordinary lineup, and it just reinforces why I'm so fond of Canada. So fond of Canada. It's really where the science leads you and what the data tell you, not what your feelings and thoughts might be about something, right? So today I'll talk a little bit about clarifying cannabis and cognition and considering the impact of recreational and medical cannabis use. It's true. I've been doing this actually for, I'm sad to say, almost 3 decades. A little over 3 decades. In terms of disclosures, I will talk a little bit about some studies that were funded by private or philanthropic donations to the MIND program, which I'll talk about in a second. We also have some funding, we're very fortunate, from our National Institutes of Health, but I won't be talking about that data. We also have some support from Charlotte's Web and Foria Praxis Ventures for specific research studies, which are also not discussed today. But, you know, better to disclose in this climate, if you watch the news, than not to disclose. The hope today is that in terms of learning objectives, among other things, you'll be able to understand some very important distinctions between those who use cannabis recreationally or for adult use versus those for medical patients. Hopefully, you'll be able to identify some changes exhibited by medical cannabis patients following 3 months of medical cannabis treatment. I was delighted to hear about your longitudinal study. That dovetails perfectly with what I'm going to show you. To explain some potential reasons for the changes observed following the initiation of medical cannabis treatment. And finally, you'll be able, probably better than I can, to summarize some clinical and cognitive changes that we've observed in patients with anxiety following 4 weeks of treatment with a novel, custom-formulated, whole plant, full-spectrum, high CBD product. We spend a lot of time in formulation in my lab. So first and foremost, I want to remind everybody, you know, cannabis has been around seemingly forever. And I'm very fond of reminding people that, are we really sure that it was a fig leaf covering Adam and Eve's most private parts? I'm not sure, because there are references to cannabis culture going back thousands and thousands of years. First reference to cannabis that we could find was really in China, about 2700 BC. Again, cannabis as medicine. In the United States, in 1850, cannabis became part of what we call mainstream medicine. That is, basically, it was adopted and added to our pharmacopoeia. That basically meant that physicians could and did prescribe cannabis for any number of conditions or indications. It wasn't a recommendation or a certification, which is what we have now in the States. It was a prescription. Huge difference. In 1935, cannabis fell out of favor. Reefer madness ensued, and by 42, it was out of our pharmacopoeia. And by 1970, it was placed in the most restrictive of the newly created Controlled Substance Act, basically Schedule 1, declaring cannabis a Schedule 1 substance, where it remains today, with very rare exception. Across our nation, slightly different from Canada, as it's really no secret, we've got medical cannabis legalized in most states at this point, starting with California in 1996. We have some states that are CBD or restricted use only, and we have a growing number of states that have legalized or in the process of legalizing recreational or adult use. Again, very, very important, slightly different from Canada's timeline. But it's important to keep this in the back of your minds collectively. Medical cannabis was sort of re-legalized, if you will, in 1996 in the United States. You're going to see why that's important in a few minutes. But considering first recreational cannabis and the impact of recreational cannabis on the brain. And for my purposes today, the lexicon I'll use is recreational or adult use cannabis will be cannabis that is used for individuals who are choosing to change their current state of being, or choosing to, quote, get high, to alter how they feel, specifically not to address a set of medical conditions or indications or even one. Important to remember that most of what we know about cannabis comes from studies of recreational consumers. In fact, young recreational users. A quick fun fact list from the US that actually is important when we think of things in the aggregate. Canada and the US have sometimes different approaches to things, but it's important for us to share these types of data because it really does factor into the decision making. Legal cannabis is still considered the fastest growing market in the US, with over 31 million people reporting what we call current use. That's use within the past month. Almost 7% of high school seniors report using cannabis daily. Teens in this country are more likely to use cannabis than cigarettes. It's over 14% now using cannabis versus those who are using cigarettes, about 4%. 65% of youth, those under the age of 18, or I think 12 to 17, don't view regular cannabis use as harmful. They don't view it as harmful. That's important. And the question then becomes, is cannabis as harmless as our youth believe? I was delighted to hear in the introduction that you're neither proponents nor opponents, and that's exactly the position I take and my lab takes. It has to do with where the data sort of guides you. So in consideration of this, when we think about our most vulnerable consumers, the important part to remember is what we call perspectives in brain development, right? Long ago and far away, the human brain was believed to be developed by the time we hit puberty, right? Sexual maturity, we must be finished. We know from pioneering efforts of folks at the NIH that the brain continues to develop throughout the second and well into, as it turns out, the third decade of life. Imagine that. Children and adolescents, therefore, are more likely to be vulnerable than older individuals, as they're in this period of what I would call neurodevelopmental vulnerability. The brain basically develops, as you know, from the back to the front and the bottom to the top. The very last part of the brain to become fully developed is the frontal cortex, just behind your eyebrows. Sadly, it's also the first part to go as we age. Biology is cruel. But I like to think of the adolescent brain as under construction, or for my cannabis consumer groups, when I give talks, I say it's half-baked. They get that? Just remember, areas involved in pretty basic functions come online first. Your limbic system, things that control senses, controlling movement, emotions, and the parts of your brain, or the part of your brain most responsible for what we call top-down control, the frontal executive system, again, critical for so many types of behaviors, are really among the last to come online. Controlling impulses, planning ahead, utilizing feedback to change your behavior, sort of the hallmarks of adult behavior. So during adolescence or even emerging adulthood, the brain is particularly vulnerable. Again, it's under construction. It isn't only vulnerable, though, to cannabis. It's vulnerable to other substances, to alcohol, to illness, to injury. Again, it's just not quite finished. When I think of cannabis and cognition and sort of the aggregate research findings, and there have been many, many decades of study here, again, predominantly looking at recreational consumers. Again, think back as to why I pointed out that medical cannabis was legalized in my country in 1996. Nearly everything we know about the impact of cannabis comes from studies of recreational consumers. They're often difficult to characterize because of inherent differences in the ways in which people use cannabis. As a result, are fairly mixed when it comes to looking at the impact of cannabis on the brain. Some factors, not all, but some, include things like sex and other demographic variables, men versus women, and for example, the developmental trajectory of gray and white matter are different for boys and girls. Frequency and magnitude of cannabis use, this is a huge one. So is product type and route of administration. Product type is very, very important to us in my group in terms of understanding, quote, what's in your weed, if you will, although it doesn't have to be weed and most often is not. But what are the constituents? Not just THC and CBD, what else is present? And more importantly, what is not in the cannabis? Exposure to specific constituents, as I just mentioned, THC and CBD. Back to route of administration for a moment. The impact of route of administration is critically important, whether they're inhalation routes, smoking or vaping, versus sublingual administration versus oral or edible consumption, transdermal or topical or transmucosal or insertion. These are all very important with regard to the outcome, that is, the effect that cannabis will have. Duration of use, how long have individuals been using? Length of abstinence at the time they're being assessed. How long have they not been using? We tend not to assess individuals who are acutely intoxicated. Never goes well for them or for the staff. But the length of abstinence from cannabis directly affects performance, especially for chronic heavy cannabis users, which I'll show you in just a second. And finally, very important to our work, age of onset of use. The age at which someone begins to use cannabis consistently, regularly. I mean this as a consistent pattern of use, not first try, a hit here, a hit there, consistent pattern of use. When I think of the neurobiologic impact of recreational cannabis use, I spend a lot of time looking at brain structure and function, as we heard a little bit about in the introduction. And in general, overwhelmingly, what we hear that studies are reporting that relative to those who don't use cannabis at all, cannabis users for recreational purposes exhibit the following: poorer cognitive performance. The most commonly affected domains, not surprisingly, memory, attention, and executive function. The figure that you see just underneath that arrow is an example of performance from a recreational cohort that is split into those with early and late onset of use. The early onset users are in the minty green in the middle there. The later onset users are dark green. And the distinction here between early and late is prior to age 16 and after age 16. The blue line are controls. And what you see, you don't have to know anything else about the data. The middle minty green bar is much, much lower than the other two bars. And this actually represents performance on something called the Stroop interference condition, where subjects have to inhibit an overlearn response tendency. They have to name the color of the anchorable word printed in a different color. So the word green, for example, printed in red. Your brain would like you to read it. Reading is the most automatic cognitive process we have. You can't help but read a word printed in your native language. And what we see inherently are, cannabis users perform worse than non-users, except when we split them into those with early and late use, it's the early onset folks driving the difference. They're statistically significantly worse at the task relative to both controls, non-users, and later onset users. We also see differences in patterns of brain activation using things like functional magnetic resonance imaging. Same task exactly, actually. This is a pattern of brain activation that's exhibited in the controls versus the smokers, the users. Again, you see a very different pattern of activation, much more posterior cingulate in the controls versus the cannabis users. But when you split the cannabis users into those with early and late use, different patterns emerge. So different pattern of activation. And finally, differences in both brain structure with regard to brain volume and changes in white matter integrity. We spent a lot of time looking at white matter in my lab using diffusion tensor imaging. I'll talk about that in a little bit. But what we see is diffusion tensor imaging allows us to look at white matter coherence, how well organized white matter is in the brain. Remember that gray matter is hardworking neurons in your brain, but then that's wonderful. But you can have all the gray matter in the world without white matter that connects brain region to brain region and allows good, clean communication between the two. You don't necessarily get the job done. So white matter integrity or coherence is very important to understand. Here, high levels of coherence or organization are what you want. And again, our minty green line in the middle of the two other bars, blue are controls, dark green are later onset users, we see significantly lower white matter organization or coherence in early onset users relative to both later onset users, and statistically significantly different from controls. Overarchingly, what we see is that higher frequency and magnitude of cannabis, greater overall exposure, as well as earlier onset of use. The earlier you begin, the greater the magnitude of change demonstrated. It's pretty straightforward. That's because the brain is vulnerable during adolescence and emerging adulthood as it undergoes these periods of critical neuromaturation. But it raises the question, what about older adults using cannabis? Would we see the same outcomes as young recreational users? And frankly, when medical cannabis was legalized in this country, in the States in 1996, you would think that people would be very, very interested in looking at exactly that using sort of longitudinal observational studies. We don't have any. I'll get to that in a second. But it's incredibly important. Why so relevant? There are more individuals that are considered older adults using cannabis than any other group. We saw a huge explosion in what we call last year use among individuals over the age of 55. It increased by about 71%, I think between 2006 and 7 and 2012 and 13. It's a big increase. For those 65 and older, the increase was exponentially greater, 250% increase. In comparison, by the way, when we're very, very concerned about our most vulnerable consumers, our adolescents and emerging adults, that group only rose about 13% with regard to rates. That's very striking. So older adults are the fastest growing group of cannabis users in the US, and it appears to be true across the globe. This is incredibly important. Newer data from 2020 shows that rates of use in those 65 and older continued to go up between 2015 and 2018. So we're not seeing things trending down, we're seeing them trending up. Again, to the point that was so eloquently articulated in the introduction, we also have reasons to be concerned about this. As much as we're very invested in understanding the ways in which we might harness the therapeutic potential of cannabis for some of our cohorts, we also need to understand the potential risks and downsides. McLean Hospital is a private psychiatric facility, and actually it's the #1 psychiatric hospital in our country. So we're very concerned about mental health conditions, psychosis, etc. Different topic for a different day. But talking about older adults, we have specific reasons to be mindful, right? First of all, as we age, our metabolism slows. Biology really is cruel. Cannabis, accordingly, takes longer to clear the body in those who are older. Modes of use. Older adults very often gravitate towards edibles or things that can be ingested. And as you likely know, that is the slowest route of administration with regard to what we call rise time, time to get an effect. You actually have to consume the product, you digest it. Again, you have processing first pass to the liver. So THC is actually metabolized into something more psychoactive, 11-hydroxy. Finally, it hits the bloodstream and the brain. And so we have a longer rise time and a much longer duration of effect relative to things like methods using inhalation. Drug-drug interactions, incredibly important. Our two primary players, and again, cannabis is comprised of over 400 constituents. Over 100 of these are phytocannabinoids, things from the plant that interact with our own endocannabinoid system, our system of receptors and chemicals. Interestingly, the two main players that everyone is focused on, delta-9 THC, our main psychoactive constituent, and cannabidiol, a primary non-intoxicating constituent, often touted for therapeutic benefit, both inhibit the cytochrome P450 enzyme system. That's an issue. Depending on the types of substances you may also be taking, conventional medications, for example, one can inadvertently increase or decrease the amount of other drugs. Then we have the regular old straightforward cognition and memory associated issues that come with aging. Age-associated cognitive decline is pretty common. We don't really have a firm understanding of the interaction between age-related cognitive decline, sort of what happens as we age, and cannabis. There has been some really interesting research, though, preclinical data that has shown poorer cognitive performance in animals may be reversed by the administration of cannabis, which is exactly the opposite of what we see in human subjects, interestingly. That study was done in 2017. Just really quickly, 3 groups of mice, basically young mice, sort of your adolescents, adult mice, and then older mice, right? So analogous to our adolescents, adults, and then older adults. And what they found was that with nothing on board, the youngest mice, not surprisingly, outperformed the adult and older adult mice on a cognitive task. Again, biology is cruel. We tend not to get better at cognitive tasks as we get older. The researchers then implanted this tiny little pump under the skin, and the animals were administered a very small dose of THC. This was not whole plant, full spectrum, anything. It was just THC. And what they found was that then the adult and older adult mice, so the 2 higher groups, significantly outperformed the younger mice who performed very poorly. So it's actually similar to what we see in terms of adolescents and emerging adults with THC products on board tending to perform worse than those without. So interestingly, it may be, the authors suggested, that the aging endocannabinoid system benefits from tiny little amounts of a tweak from cannabis. So despite these types of compelling preliminary data, there's been virtually no studies systematically addressing the impact of cannabis use in older adults, which is stunning. The potential risk of cannabis use disorders among medical cannabis patients, just in general, or the efficacy of medical cannabis use for various indications. Typically what we see for medical cannabis studies in our country are acute administration for single types of conditions and indications. There's no longitudinal data available until We'll get there. One other quick reminder, medical and recreational cannabis use should never be considered the same. There are a lot of overlap, a lot of similarities, and many areas that overlap between recreational cannabis consumers and medical patients. For example, products chosen. Very often, it'll be the identical product. Routes of administration are often sometimes common, although I will tell you, I've never heard of a recreational consumer, not yet, using a suppository. I'm sure I will. I haven't yet. But really importantly, goal of use is this, in my mind, one of the single deciding factors here. When the goal is to feel high, to change one's state of being, to feel euphoric, that's typically recreational or adult use. When the goal is to alleviate or address specific symptoms, I hear from my medical patients, look, I don't want to be high. I want to be able to drive my car. I want to sleep through the night. I want to take a walk in the park with my husband. That's very different. That's a medical cannabis patient. The goal of use often drives the constituent profile of the products that these folks choose, not surprisingly. It is not a shock that our recreational cannabis consumers typically choose products high in THC. Again, they're looking for that. And the proliferation of things like concentrates in our country and really across the globe, products that start life at about 35 or 40% THC and go north of 95% THC with virtually undetectable amounts of CBD inherent because they're designed to give the user a big bang for the buck, if you will, with regard to THC. That's what those folks often choose. Not always, but our recreational consumers are typically interested in THC. Our medical patients may choose products with considerable THC content, but they're also much more likely to choose products with a wider array of cannabinoid profile, a much broader profile, if you will, including things like CBD, but other non-intoxicating constituents, the so-called minor cannabinoids that often have a major effect. Age of onset is also very different. Typically, we see use initiating during adolescence or emerging adulthood for rec use, and our medical cannabis patients typically are over the age of 25, with the exception of the pediatric onset intractable seizure disorder kids that Oren will talk about, I think, tomorrow. So important distinctions. So I mentioned 1996 as an important time. The US first legalizes or re-legalizes cannabis. And you would think that we would be collecting data like crazy since that time. But I will tell you that when I scoured the literature after cannabis was first legalized here in Massachusetts in 2012 and could find no studies, longitudinal studies of the impact of cannabis on things like cognitive performance, clinical state, sleep, quality of life. I couldn't find anything. So I started the MIND program, Marijuana Investigations for Neuroscientific Discovery. It was called MIND and not something with cannabis in the title because here in the States we have medical marijuana laws. We don't have medical cannabis laws. So it's, I don't love it, but anyway, now we have this branding, so we can't change it. It's basically the first ever longitudinal study in the US of medical cannabis patients and the first to track cognition as a primary variable. First also to use neuroimaging techniques, and finally the first to have whole plant, full spectrum clinical trials for folks with anxiety. That was our first IND. In our country, we have to have an investigational new drug approval before we can use a formulation in a clinical trial model. We also have INDs for a number of other indications. So I was very happy to hear about your research projects because I think there's a lot of overlap, which is wonderful. But basically, this program is dedicated to looking at the impact of medical cannabis on these different things: cognitive performance, clinical state, conventional medication use, quality of life, measures of brain structure and function, sleep, sex, you name it, we're looking at it. We use longitudinal, observational, cross-sectional survey, and finally clinical trial models. And we support a number of projects designed to look at custom formulated cannabinoid-based therapies for a range of indications and conditions, including anxiety. I like to say our MIND program is on a mission to support these range of studies that will generate, again, to your brilliant introduction, ecologically valid, empirically sound data in order to close the gap between policy and science, and look at the unique and synergistic effects of cannabis and its constituents, to look at the efficacy of cannabinoids for specific indications and conditions, and at the same time being able to assess and potentially mitigate risk and harm. We also want to document and prevent patterns of misuse, of course, and clarify the overall impact of cannabinoid treatment on physical and mental health. The goal is really to harness the therapeutic potential while minimizing harm of treatment. That's the goal. So I'm going to show you some data from our ongoing study. Our first study was an ongoing longitudinal observational study, and it's still going. This study is designed to measure all those things I just mentioned, but very importantly, we collect all of this data in our patients before they begin using cannabis. I can't stress the importance of this enough. So folks are given a certification or recommendation for cannabis use, no prescription here, very important. But before they begin using, they actually come into the lab. And we actually ensure that they are free of cannabis use using a very comprehensive screening approach and, of course, testing to make sure that there's no urinary cannabinoids. The study is designed to follow patients for up to 2 years of treatment at this point. We started with 1 year. So we see them at baseline, no cannabis on board. And then after consistent use of cannabis for a minimum of 3 months, 6, 12, 15, 18, and 24, we have monthly phone check-ins with them. I have an extraordinary small but very hardworking staff, where the patients actually keep track of what they're using, where they got it, how much they're using, what the label says, and when they settle on a regimen that works for them, we actually have the most commonly used products sent to an outside lab for cannabinoid constituent profiling. Because it's not a clinical trial, and I'm not giving them the product, having a lab result, a certificate of analysis, tells me, quote, what's in their weed. We also calculate absolute exposure to cannabinoids. We quantify it using information from the lab analyses, the certificates of analyses from points of purchase, and then finally product labels, because sometimes that's helpful. In order to get into the studies, they have to be over 21 and certified for treatment or plans to use a product that in the States do not require any kind of approval, the so-called hemp-derived products in this country. Anything with less than 0.3% THC by weight is deemed hemp. Hemp was approved for use in this country in 2018. Some argue it really was approved in 2014, but a different story here in the States. But the long and short of it is they have to have approval or certification or not need one. And they can use for any variety of indications. I wasn't sure how many people would get when I started this, so I didn't limit it to just pain, just anxiety, PTSD, etc. And they must not have used cannabis regularly in the past 12 months. You'll see, though, it's much, much longer in general. They have to, if they test positive for urinary THC at baseline, or have a lower IQ than 75, or a history of psychotic disorder or neurologic disorder, really any serious medical condition, they're actually excluded. So I'm going to show you some data on the first 55 patients who completed a baseline and at least one follow-up visit. In general, these folks are well-educated with over 16 years of education. They're about 49 years of age, so well beyond the period of neurodevelopmental vulnerability. We're going the other way at that point. Predominantly Caucasian. And their duration of cannabis use, duration of cannabis abstinence at the time of their baseline was approximately 23.5 years, so a long time. Most of these people report a very remote history of cannabis use, typically in college or the end of high school. Why are they using? The most common indications in this country are pain and anxiety. So, not surprisingly, some are using for pain, anxiety, or PTSD, sleep, mood, attention, and other. What's other? Inflammatory bowel disease, migraine headaches, things like this. I mentioned that we have this quantification, this metric where we quantify exposure to THC and CBD, and that's been incredibly helpful. There wasn't one that existed. We created one for use in the lab that seems pretty accurate. And what I can tell you, and what you can see really, is that between baseline and 3 months, or actually clearer here is 3 months, 6 months, and 12 months, you can see that our patients are predominantly using a higher CBD than THC products. Not surprising, right? But interestingly, you can also see that the amount of THC that they're using on average goes down over time. We hear this a lot from patients. I don't need as much. Interesting. The same also appears to be true in CBD. People tend to find what works and use the lowest effective dose, which is what we're looking for. We also have, at this point, I'll show you data on the first 19 treatment as usual patients. So these are not patients who are using cannabis, but have similar conditions or indications, medical conditions, as those in the treatment group, but who are not using cannabis. So first, looking at conventional medication use. We're in the midst of this horrific opioid crisis, and people are very invested in understanding the potential role for cannabinoids. Since many patients don't get full symptom relief from conventional opioid-based therapies and often complain tirelessly about the countless side effects associated with opioid use, we see more and more patients trying to find other ways. Medical cannabis states in the US have reported fewer prescriptions for opioids are issued and fewer opioid overdoses than states without fully legalized medical cannabis, not the CBD only states, but fully legalized medical cannabis states. But a more recent study raised some questions about those findings. and really underscored the importance of looking at what we call individual level data. This is as individual level data as one gets. First looking at these patients, what we saw was that our medical cannabis patients using conventional medications reported a rather notable decrease in their use after only 3 months of medical cannabis treatment. We saw a 13% reduction in the use of opioids, a 22% reduction in the use of over-the-counter analgesics or pain relievers, and a 39% reduction in the use of benzodiazepines. Those reductions continued at later visits. So that's very, very important. Remember, this isn't the study doctor saying, hey, now that you're using cannabis, you should cut down. This is all on their own. So that's important. Turning to cognitive and clinical assessments, first clinical. When we look at mood, very, very importantly, we're trying to assess these things at baseline and then follow these folks over time. And what we see are things like the profile of mood state, total mood disturbance. This is just a total score. The green line demonstrates the medical cannabis patients. The blue line is our treatment as usual group. And what you see is between baseline and, let's say, 12 months, a striking reduction in total mood disturbance scores over time. We don't see the same reduction in the treatment as usual patients. The same is true for the Beck Depression Inventory, a really classic, very commonly used self-report scale of depression and depressive symptoms. Again, self-report, striking reduction in the medical cannabis patients, 12.6 at baseline to 6.2 after 12 months. We don't see the same reduction in treatment as usual, folks. Looking at anxiety, a very important area for us using the Beck Anxiety Inventory, again, statistically significant reductions between baseline and 12 months. We don't see in the treatment as usual, folks. Frankly, we have a measure of state and trait anxiety as well. We don't see the same clinical improvement in the treatment as usual, folks. We do see clinical improvement that reaches statistical significance in the medical cannabis patients. Sleep. Sleep is one of those domains that actually cuts across absolutely every condition and indication, no matter what it is. We often see sleep disturbance, whether it's mood-related, chronic pain, all sorts of things wind up impacting sleep. So very often we see changes in the sleep scores before anything else. And this data is the PSQI, the Pittsburgh Sleep Quality. And what we see here, again, a statistically significant improvement in sleep quality in our medical cannabis patients. We do not see the same in treatment as usual, folks. But here, the scores, lower scores are better here. We go from an 8.9 to a 6.3. Pretty striking. When we consider measures of cognition, and I'm just going to focus primarily on frontal executive tasks, as I mentioned, we have things like that Stroop color word test that I explained, the trail making test, which basically has two parts, a psychomotor speed task, and then a second task that is an alternating set demand. So subjects have to connect dots alternating between number and letter. So 1A, 2B, 3C, it's an executive task. Wisconsin card sorting task, often considered a gold standard measure of cognitive performance, specifically frontal executive function. 4 cards are placed in front of a subject. They're given a deck of cards and told with nothing else, take the card and decide in front of which of these 4 it belongs. We'll tell you whether you're right or wrong. There's different ways to sort the cards. What they don't know is that after a number of correct consecutive responses, the rule changes and they have to utilize that feedback to change their behavior. Classic frontal executive tasks. Letter number sequencing, subjects get a mixture of numbers and letters all jumbled, and they have to repeat the numbers in order and then the letters. And finally, phonemic fluency. Name for me as many words as you can in a minute that begin with this letter, or this letter, or this letter. So looking at the cognitive data is a little bit different for our clinical data. What we see here, again, over time between baseline and 12 months, are some interesting improvements. If we look at Stroop interference time, our patients seem to get faster, but actually so do our treatment as usual folks. So that was kind of interesting. For the Wisconsin card sort categories and perseverations, again, that classic frontal function assessment, we see statistically significantly better performance between baseline and 12 months. We don't see that in our treatment as usual, folks. Perseverations, repetitive incorrect responses. People will throw a card that meets for color, they'll be told no. They'll throw a card, again, it meets for color, they'll be told no. That's a perseveration. An inability to utilize feedback to change your behavior and look for the next correct rule. Statistically significantly improved performance, significantly lower perseverations between baseline and 12 months. In our medical cannabis patients, we don't see that in our treatment as usual folks. They do get a little bit better over time, but again, not at that 12-month mark. Letter number sequencing, again, we see a lot of improvement in the medical cannabis folks. a little blip of improvement in our treatment as usual, folks. And really the same for phonemic fluency, nothing hugely different. So pretty interesting. What we're not seeing is a huge level of degraded performance in our medical cannabis patients who, remember, are naive at baseline. So very often people think, well, if you're going to add cannabis, they're going to get worse. Just like our recreational consumers look worse than those who don't use. We don't see that here. When we turn to measures of memory, verbal learning and memory in particular, we have a measure called the Ray Auditory Verbal Learning Test that allows us to track the ability to learn over the administration of a serial list 5 times. The list is repeated. Subjects are asked to repeat the list after each time, and then a short and long delay are interspersed. And we use alternate forms to prevent practice effects. What we see here is almost identical performance over time. But again, I would tell you, our medical cannabis patients aren't getting worse. They're not getting worse. They're holding pretty stable for both learning and memory measures. So that's important. When we think about neuroimaging, or the ways in which we can look at sort of the neurobiologic change over time, there's lots of different ways we do it. One of the tasks that we adapt for use during functional magnetic resonance imaging, and I should say our lab is a one-stop shop. One trip to the scanner buys you structural imaging, functional imaging, spectroscopic imaging or in vivo brain metabolite ratio assessment, and diffusion tensor imaging, which I'll show you as well. So one visit, lots of different type data. So multimodal imaging is really important to us. This task, the multi-source interference task, is designed again to be that classic frontal task of inhibitory function. Basically, it goes like this. There's 3 numbers that appear on a screen, and there's 2 conditions. During the control condition, there are distractor numbers and a target number. The subjects are simply told, tell me the identity of the number that's different using a button box. Okay. During the control condition, the 2 distractor numbers, the non-target, are zeros, and the target is placed in its correct position as on the button box. So in panel 1, the answer is 1, and obviously 1 is the first button on the box. On the second panel, the answer is 2, the third is 3. Pretty simple. In the interference condition, the distractor numbers are not zeros anymore, and now the target number is not placed in its appropriate position. So remember, the subject has to identify the identity of the number that's different and inhibit the visual position. So in that first panel, 2, 1, 2, the answer here is 1. Subjects really want to hit 2 because it's in the second position. Second panel, the answer is 2, but they want to hit 3. And in the third panel, the answer is 3, but they want to hit 1. You can see, inhibit the position and instead tell me the identity. What do we see? When we look at performance, both the medical cannabis patients and our treatment as usual folks actually show improvement over time. Faster response times and improved accuracy in terms of the discrete task performance. Kind of interesting. So that's one thing. We have not published these data. These are actually under review now. When we look at the pattern of activation, though, and then the association of the activation patterns with performance, it's a slightly different story. So here's what our medical cannabis patients look like at baseline. And this is a group of 29 folks in the cingulate cortex region of interest. You can see that yellow blob right in the mid-cingulate. And the frontal region of interest, we see bilateral activation. What happens, that's at baseline, during the performance of that interference condition? What happens at 3 months? Whoa, remember, their performance got better, but they're using less cortical resource to do it. Think of it just as nothing more complicated than the number of voxels activated, voxels that meet a statistical threshold, for example. In baseline, it's 125 in the cingulate cortex region of interest. 3 months, it's only 44, and their performance is better. So less cortical resource to get a better job done. Same is true in the frontal region of interest. When we look at baseline to 6 months, slightly smaller sample, but I don't want to cheat and pretend it's 29. This is 20 people. We want to actually use their own baselines. Again, same pattern, more activation at baseline than at 6 months. So that's interesting. When we take them together and look at the patients versus the controls, what we see is that our medical cannabis patients show decreased frontal activation, less activation in the frontal cortex that accompanies better performance. Treatment as usual, folks, are showing increased frontal activation. See on the right, baseline in the first row there, first column, baseline in the cingulate and the frontal, 0 voxels and 199, and then flip to the 3 months, 18 and 487. So opposite pattern, more voxels required to get the better job done. So again, medical cannabis folks, decreased activation, better task performance. Treatment as usual folks, increased activation to get better performance. So they have to work a little harder from a neural perspective. Diffusion tensor imaging is one of those imaging techniques that allows us to look at, as I mentioned, white matter fiber tract integrity, and we have different ways and different areas that we measure. This is just a quick fractional anisotropy image. So there's basically a couple of outcome measures from DTI. One of them is fractional anisotropy, or FA. And lower numbers here, the numbers go from 0 to 1, and we want higher numbers. Higher numbers represent greater white matter fiber tract integrity. So if we look at the data from our medical cannabis patients versus our treatment as usual patients, we see some rather striking changes. And I will tell you, full disclosure, when you do multimodal imaging, you do the same protocol at baseline, at 3 months, at 6 months, at 12. I didn't expect to see anything with regard to white matter. Not at 3 months, not really. What we're seeing is, if you look at that, again, the top section, the medical cannabis patients, that white bar is baseline. The minty green is 3 months, that dark green is 6 months. We see a statistically significant increase in white matter coherence or organization between baseline and 3 months and baseline and 6 months in our medical cannabis patients. So we're seeing a structural brain change. We do not see the same pattern in the treatment as usual, folks. If you look at the bottom, we see one blip of increase, and that doesn't really hold. It's the right GNU. We're seeing it in every region we're analyzing in the treatment patients. So medical cannabis patients are exhibiting these increases in white matter organization over the course of treatment, which are not seen in our treatment as usual group, suggesting that at least for some, medical cannabis may be related to structural brain changes in a pretty great direction. We'll leave it there. So to summarize this data, after 3 to 12 months of medical cannabis treatment, our clinical data show areas of significant improvement. Mood, anxiety, sleep, quality of life, definitely different from the treatment as usual, folks. The patients are generally also showing improved performance on measures of executive function and stable memory performance. This is pretty important given what we know from our studies of recreational cannabis users that have demonstrated decrements in executive function compared to those who don't use. We also see notable decreases in the use of conventional medications, for example, opioids, benzos, and analgesics. Very, very important. In terms of the imaging data after 3 months of treatment, we also see this potential normalization of brain activation during frontal executive tasks, which accompanied improved task performance in our patients. And our DTI data suggests increased white matter coherence between baseline and 3 and baseline and 6 months, suggesting these structural brain changes may be related to the medical cannabis use. Overall, in general, I'd say in clinical, cognitive, and neuroimaging measures, the same degree of improvement or changes were not observed in our relatively small sample at this point of treatment as usual patients. Again, same types of patients with regard to the conditions, but they're not using medical cannabis. But taken together, these data really suggest that medical cannabis may be a safe alternative or adjunctive treatment for at least some patients. So some people ask, well, why do we think they're getting better on these cognitive tasks, for example? Symptom alleviation. Patients who are taking, for example, for chronic pain say, I feel better. I can think more clearly. When the nociceptive center of the brain is not processing painful stimuli all the time, there's more resource to be allocated for things like, I can remember my granddaughter's phone number. Medical cannabis product choice, as I mentioned. Unlike recreational consumers who typically choose products with considerable THC content, our medical cannabis patients are often choosing products with a very rich and varied cannabinoid profile. CBD has been shown to potentially mitigate or reduce some of the less desirable negative effects of THC. That may be part of it. Certain other cannabinoids may also directly or indirectly affect cognitive performance. We don't know yet. That's sort of an area that is desperate for more work. The reduction in the use of conventional medications is also really important. We've heard a lot from our patients saying, I have this cognitive hangover feeling after I take Xanax, for example, or my opioids. When you reduce that, it may be the kind of thing where people feel a little bit clearer with regard to cognitive performance. And finally, age of the consumer. Medical cannabis patients in our current study were 49, well past adolescence or emerging adulthood. And our early onset recreational consumers typically are the ones with the worst cognitive performance. So we may be looking at that. All of this is to be explored with ongoing research, all of it. We have a number of ongoing additional studies. We have these quasi-clinical trials of veterans using cannabis products. We call it Serving Those Who Have Served. The goal is to look at the impact of cannabinoids on clinical state and cognition in our veterans. We call it Serving Those Who Have Served. Like I said, multiple cohorts, cannabis naive, and also cannabis using people. They actually all start using one commercially available product, which is a godsend. We have a women's health initiative at Mind that's designed specifically to look at conditions and indications that either disproportionately or exclusively impact women, things like menstrual or menopausal related issues. I have a study that looks at medical cannabis in older adults with pain and opioid use, to look at the impact of medical cannabis use on a number of things, including the changes potentially in conventional medication use. And we have a number of clinical trials either underway or pending. Underway, I'm going to show you data in just a second. We have approval for pain. We have an agitation, anxiety, and Alzheimer's-related dementia study ongoing, and anxiety, or what I like to call high anxiety. Just kidding. Very quickly, we have the very first clinical trial of a whole plant full spectrum product for individuals with moderate to severe anxiety. It's an open label to double blind trial design. So there's 4 weeks of active treatment, and the open label phase was really designed for us to make sure that the dose was correct. Again, it's never been done. This is a whole plant full spectrum product that wound up being it was not my first choice, this particular one that I created, long story, but it wound up producing a product that I was very happy with. And again, I had to formulate it myself, 10 mg per mil of CBD at a 3-time-a-day dosing, but it is a whole plant full spectrum product. So it's about 0.22 to 0.24 mg of THC per serving, as well as cannabichromine, cannabinol, cannabigerol, etc. It's a sublingual administration, and again, it's a full spectrum solution. A quick look at the study timeline. We assess everything we can think of over the 4 weeks of treatment, including anxiety and mood scales, sleep, quality of life, and other rating scales are done at baseline and after 4 weeks of treatment. We assessed their urine at every visit and I'm very glad we did that. And we did a small PK/PD study in the open label folks as well. The aims were really to verify the correct or adequate dose and to look at clinical ratings of anxiety, mood, sleep, and quality of life over this 4-week period, and to look at whether or not we saw any changes in cognitive performance. Ultimately, we were also trying to figure out the safety profile and the potential for serious adverse events. And I was particularly interested in whether or not we'd see THC positive status over time, because so many across this country and yours and the globe are convinced that if they use a hemp-based product that you can buy in a gas station, they'll never be positive on the drug screen. Really quickly, it was 15 folks. We bumped one, I won't go into it, but 14 completed, and they were those who met for moderate to severe levels of anxiety. Here's their baseline scores. The study drug was extremely well tolerated. We had no serious adverse events, and no patient ever reported any intoxication. On average, they used about 3.5 mils per day instead of 3. We weighed the bottles on the way out, we weighed the bottles on the way in, and despite getting a graduated dropper, people are people and not rats. We're not administering it, they are. So it looked like they were using a little more than they should have, but that's great. That really is equivalent to about just under 35 megs of CBD and just under 1 milligram of THC per day. Really quickly looking at results in terms of anxiety. That's what this was created for. So the Beck Anxiety Inventory, the OASIS, and the HAM-A, all measures of anxiety. We see really striking decreases. Again, it's an open label phase. But remember, placebo responses are typically somewhere between 20 and 25%. 79% decrease in the BAI, 20.2 to 4.0. OASIS, 11.2 to 3.3. That's a 70% decrease. And on the Hamilton Anxiety Scale, just under 20 to just over 3. So pretty amazing. When we look at mood ratings, the BDI or Beck Depression Inventory, we go from a 20 to a 4.4. That's a 77.9% decrease, very striking. And again, that profile mood states, that total mood disturbance, higher numbers are worse. We go from 65 to a 24, 63.6% decrease. So they're definitely reporting feeling better on self-report and examiner-driven scores. When we look at sleep and quality of life, that PSQI, that sleep measure, goes from a 9.2 to a 5.8. That's a 36.4% decrease, very striking. We also see real improvements in the SF-36, a measure of quality of life across a number of domains in terms of energy and fatigue, emotional well-being, social functioning, etc. A lot of significant improvement here. Again, self-report. Turning quickly to cognition, we saw stable or improved performance between baseline and 4 weeks of treatment. More correct responses, faster times, and fewer errors. So again, we don't see any cognitive decrement in our patients after initiating this custom formulated whole plant full spectrum product, quite the opposite. It is also true for the last 2 measures, the letter number sequencing and phonemic fluency. Again, more correct responses, faster times, and fewer errors between baseline and 4 weeks of treatment. I mentioned THC positive status. Very few studies so far have looked at whether the use of these full spectrum high CBD products might yield a positive result on a drug screen. So we assessed them with in-house assays at every visit. We also sent it out for quantification of urinary cannabinoids. And we looked at potential relationships between those who are THC positive and demographic variables, creatinine, reflecting kidney function. Really, really interesting. After 4 weeks of treatment, exactly 50% of my sample, 50% of the patients were positive. Really very interesting. The GCMS results confirmed the assay findings and also indicated that we had positive results, even if the GCMS indicated the metabolite levels were below the assay's actual lower limit of detection, likely due to cross-reactivity with other cannabinoids. But be aware, if you're using a whole plant full spectrum product, you may actually be able to be detected on the drug screen. THC status was only associated with urinary creatinine levels. We didn't see anything, for example, with body mass inventory, I thought for sure, or amount of product used. We had one subject who used way more product than she should have. That person remained negative. So it wasn't amount of product or BMI. Patients consistently using these full spectrum hemp-derived products then may test positive for cannabis use on a urinary drug screen. Very important take-home message. So overall, our clinical trial, the open label at least, suggests that the use of this custom formulated a whole plant derived high CBD product results in less severe anxiety and fewer anxiety related symptoms after 4 weeks of treatment relative to baseline. Self-reported improvements in other mood symptoms, sleep and quality of life were also noted, could be related to reduced anxiety. We also saw improvements on tasks of executive function and no serious adverse events reported at all, or intoxication. That's great. A definitive assessment of the impact of this treatment will be ascertained during the double-blind phase, which is now underway. I'm very excited. We also need studies with larger sample sizes to more thoroughly assess which variables, product use, BMI, age, sex, race, medication use, etc., contribute to the positive THC status, which we saw in some folks, but not all, particularly those with higher creatinine levels. And importantly, the study product that I used, if you calculate it, that I created, had 0.02% THC by weight. Hemp products can have up to 0.3% THC by weight. That's more than 10 times the allowable amount of THC. So my product, again, 0.02, the allowable amount by law, 0.3, 10 times that. So if you take these results and think about what that could mean, I think it's clear. It's often assumed that folks using these products will be negative, but this really may not be true. And this underscores the potential for adverse consequences when we think about this, right? Loss of employment and legal or treatment ramifications. People who are in treatment who say, I'm not using anymore, I'm just using CBD. Likely it isn't just CBD. Single extracted compounds in these products, unless they're pure isolates, are hard to find. And I've looked at, I don't know how many thousands of lab reports from companies that say it's a pure isolate. It's not a pure isolate. So really, really important. Important take-home message, medical cannabis patients and recreational cannabis users are not the same. We know that THC is often associated with negative cognitive and clinical outcomes, but our medical cannabis patients who are often using products with more varied profiles appear to exhibit improvements in a number of areas, and they certainly aren't exhibiting huge decrements. That's hugely important. These improvements in our patients may be related to a later onset of use. The brain's no longer so vulnerable. Primary symptom alleviation, I feel better, so I perform better. Reduction in the conventional medication use, or a number of other factors, all of which need more research. And it's really important to remember that real-world data from longitudinal studies of medical cannabis patients are critical, since additional clinical trials assessing multiple product types and modes of use really get their best info from these types of studies. We need clinical trials to assess additional domains and to examine the impact of specific cannabinoids, either on their own or in combination with other cannabinoids and constituents. I'm actually doing a separate arm of that open label to double blind phase where we had that whole plant full spectrum product. It's a high CBD product, but it's full spectrum. That same exact thing with everything else pulled out, but the CBD from the same chemovar, the same cultivar, so the same source. That's virtually impossible to find in the literature, but I think we need to know, is there really a true entourage effect? We think so. And with that, I would thank you for your time and attention and acknowledge my extraordinary staff and my collaborators and take any questions you might have.
Thank you very much, Dr. Gruber.
Sure.
That was a wonderful talk. We do have a few questions in the Q&A area. For the attendees, if you have other questions, you can type them in, and if we run out of time, then perhaps Dr. Gruber can stick around and type in the answers as we go on break and into the next session. So one of the questions from Kelly Smith, the study lab tested the cannabis that participants were using and recorded what symptoms they were targeting. Was there a correlation/comparison made of strains and cannabinoids that were successful in treating specific symptoms or worsening?
So it's a great question, Kelly, and I feel like I should give you $50 for having asked them. That's the goal of collecting the actual constituent information. And I'll tell you, I took a lot of, I'll use a clean word, heat for doing that, because it's not legal in my country to do this. If I have a Schedule 1 license and can administer, and congratulations to you guys for also getting your stuff in line. Believe me, I know how hard it is. But recreational products, actually, or products that people are using on their own, I am not able to do anything with. So that was hard to do. We are not yet at the stage where we can make statements, I think, about specific, quote, strains. To me, it's much more important to understand the constituent profiles. Remember that strains are based on almost nothing other than one's desire to call something something, right? So, for example, let me think. Malibu OG is very different here in Massachusetts than it is in California or Tuscaloosa, Alabama. So for me, it's a little bit more important to understand the actual constituent profile versus the actual, quote, strains. That's really what we're looking for. I hope that's helpful. I know people are desperate, like, oh my God, what about Blueberry Kush? What's that gonna do? I would love to answer your question. And when we have hundreds of thousands of data points, maybe we can. I also think it's tough because so many of these things change over time. When you do a clinical trial with cannabis that is harvested, you want to lock out your, quote, lot, right? Because there are differences over time in terms of the different factors that weigh into the actual cultivar, the plant itself. So very, very important. We think that getting the information on the constituents in the products is our best bet.
Great. We'll take one more question here from an anonymous attendee. Why are medical cannabis patients and TAU patients different at baseline in terms of cortical activation?
They're not actually, they weren't statistically compared at baseline. Just so we're clear, they're actually only compared to themselves baseline and 3 months or baseline and 6 months, depending on the contrast. And that's because the TAU group is much smaller than the current medical cannabis group. It's a great question. But we actually go ahead and use just the group of folks that we have to do the contrast. I think it looks very sexy to do these comparisons to each other, but it's not statistically appropriate to do it yet. We can't do that. We want everybody matched. We're getting there. We're pretty much there. And we actually have just done a subset analysis. So we took all of our TAU folks and matched them with their, quote, medical cannabis cohorts. So that's a different question. I expect that they actually will be matched at baseline.
Great. James or Jason, do you have any other comments? You're muted, James.
Oh.
I'm muted, sorry. I just want to say thank you so much. That was a really inspiring talk by how many different methodologies were brought to bear. Very exciting work. Thank you so much for joining us.
Well, thank you. It's an honor to be here, and congratulations on all the great work, really. Thank you so much.